Hi everyone, my name is Gustavo Elizalde. Today I have the pleasure to introduce to you our new research article, which is about the toxic effects that environmentally relevant concentrations of fluconazole may pose to Danilo embryos. Okay, so for those who have not heard about fluconazole or are not aware about the impact that this drug has in aquatic ecosystems, I will give you a brief introduction. Fluconazole is a fungicide of the azole family that people use in dermal and oral medications to treat fungal infections. Nonetheless, we can also find it in household products such as soap, shampoo, dermal creams, shower gels, and toothpaste. After fluconazole is administered, it enters the sewer system, passes through wastewater treatment plants, and reaches the aquatic environment. Although few authors have evaluated the occurrence of fluconazole in water matrix, it is noteworthy to mention that this antifungal drug has been found in different water bodies around the world, such as wastewater, surface water, and even drinking water. Thus, we believe it is important to keep studying the occurrence and distribution of this drug in the water matrix. As fluconazole is present in the aquatic environments and it inhibits the cytochrome, it may cause non-desired effects in non-target organisms or aquatic species. Up to date, only 3 studies that we are aware of have evaluated the toxic effects of fluconazole in aquatic species. Nonetheless, in all of them, the authors did not use environmentally relevant concentrations. Taking into account this information, we aim to study the embryotoxic effects that fluconazole may induce in freshwater Danio. For this effect, we expose Danio embryos to 9 environmentally relevant concentrations of fluconazole and evaluate the mortality, malformations, and hatching rate. Moreover, we also aim to determine whether or not these concentrations of fluconazole may disrupt the oxidative status and gene expression of Danilo embryos. According to our results, all concentrations of fluconazole induce pericardial edema, yolk deformation, down deformities, scoliosis, and delay in the hatching process. Here, it is noteworthy to mention that the delay in the hatching process was in a concentration-dependent manner. Therefore, we found that less than 20% of the embryos exposed at the highest concentration of this drug have hatched at 72 hours post-fecundation, and only 78% of them achieved to hatch at 96 hours post-fecundation. Concerning oxidative stress, we found this drug altered the redox equilibrium of fish, promoting the production of lipid peroxidation, hydroperoxide, and protein carbonyl content in a concentration-dependent manner. Even though we also found that the activity of superoxide dismutase and catalase were also increased, our integrated biomarker index demonstrates that the embryos exposed to fluconazole were not able to overcome the overproduction of oxidative stress. Finally, regarding gene expression, fluconazole altered the expression of several genes related to organogenesis in the early life stages of development. Here, it is important to point out that the downregulation of these genes could be related to increased production of retinoic acid, as it is implicated in different signaling pathways related to embryogenesis. Thus, we believe that malformations observed in this study could be related to fluconazole capacity of boost the production of retinoic acid. Collectively, our results pointed out that fluconazole at low concentrations may alter the embryogenesis, oxidative status, and expression of several genes in the neuroembryos via an impairment in sterol and retinol biosynthesis. Thus, our results provide some of the first evidence that fluconazole, even at environmentally relevant concentrations, is harmful to aquatic species.